Good afternoon, everyone. Welcome to a special Wednesday afternoon lecture. This one, the Astute Clinician Lecture, where we seek to bring individuals who are combining scientific expertise with clinical expertise and making observations that transform our understanding of health and disease. And today's speaker, Dale Abel, is a great example of someone who has spent his career doing just that. Some of you are probably here a little anxious about whether the government's going to shut down tomorrow night, but for the next hour, just put that all aside. And it looks as if we might squeak by this time, although there's a few steps to happen between now and midnight tomorrow. And some of the rest of you are probably dreaming of Valentine's Day because it is, after all, a week from today. So the fact we're talking about the heart, not such a bad thing. And the fact that some of you are maybe planning to give chocolates to your loved ones, well, lots of sugar. Well, think a little harder about that after this presentation, about whether that's really a good gift or not. We'll all learn something along the way. Our speaker has a remarkable trajectory in terms of his training and his contributions in various academic institutions. He received his MBBS at the University of the West Indies in Kingston, Jamaica, and then a Ph.D. from Oxford University. He was a chief resident in internal medicine at Northwestern. He spent a little time in Boston at a place called Harvard, and then moved to the University of Utah for 13 years, where he moved through the ranks from assistant to full professor and medical director of the Diabetes and Endocrinology Center. But since 2013, he's been at the University of Iowa, initially as director of the Division of Endocrinology and Metabolism, and now as the Francois Aboud Chair in Internal Medicine, as well as Department Executive Officer, which means he runs the place. So he obviously must have skills not only in research, which is what he's going to tell us about, but also in a way of managing a complex and critically important part of a very important institution right there at the University of Iowa. He's a former Rhodes Scholar. He's received a number of important awards. I mentioned particularly the Gerald Auerbach Award. He has been elected as the president of the Endocrine Society. We are grateful to him because of his contributions to NIH, because he has served previously as the chair of the Board of Scientific Counselors for NIDDK, and is currently a member of an advisory committee to NHLBI and a long-term grantee. He's been elected to the ASCI, the AAP, and the National Academy of Medicine. And his current research interests, which he will talk to us about, very much play out in this understanding of the role of sugar and insulin in the dysfunction in the heart that happens in diabetes. So please join me in welcoming Dr. Dale Abel.
Thank you very much for turning out in this tremendous snowstorm that has closed down Montgomery County. And having come from two places where there's real snow, I always find it very, very interesting how the rest of the world reacts to the threat of snow. Nevertheless, I'm going to spend the next 45 minutes or so speaking to you about some work that we have been doing over the years, really trying to understand the mechanisms that relate to cardiovascular complications of diabetes. Now, it shouldn't be lost to, I think, most people in this room that we have really seen a tremendous increase in the prevalence of obesity and diabetes over the last 2 or 3 decades. And in fact, this is a slide that I know that Griff has used quite a bit in his presentation, showing that really there's been an exponential increase in the number and the percentage of people with diabetes in the U.S. I think it's also important to emphasize that diabetes really is a global epidemic. And even although we think about the numbers in the U.S., just note that in the developing world, sub-Saharan Africa, for example, and in the Middle East, you're seeing double and triple-digit increases in the expected incidence and prevalence of diabetes between now and 2045. Now, the reason why diabetes is something that we really need to worry about are its complications. And when I was in medical school, we learned about the microvascular complications of diabetes, so retinopathy, a leading cause of blindness, nephropathy, a leading cause of renal failure, neuropathy, a cause of pain, limb loss. But really, the elephant in the room is cardiovascular complications of diabetes. And oftentimes, when we think about that, we think about atherosclerosis, MIs, and stroke. But I'm going to suggest to you today that there's another diabetic complication, which is heart failure. And I think that's largely unrecognized. But I hope to kind of show you in the course of this talk why that needs to be changed. This is a classic study done almost 2 decades ago, looking at about 10,000 individuals and looking at both the prevalence and incidence of heart failure across the lifespan of individuals with type 2 diabetes. And you can see very clearly that at all ages, there is a significant increase in heart failure incidence in individuals with diabetes. Roughly a 2-fold to 3-fold increase in either the incidence or prevalence of heart failure among individuals with diabetes. What was very interesting in this study, they actually looked for what could be potential predictors of heart failure in this population. And some of them were not unexpected, such as ischemic heart disease, hypertension, or renal failure. But very interestingly, and this has really held true, is that if you are diabetic and you are a woman, then your risk for heart failure is actually twice as high as the risk for heart failure if you're a diabetic man. And we still actually don't fully understand why that is. The other interesting thing that came out in the study is that if you are an insulin user, then you also are at a somewhat greater risk of heart failure in diabetes. And this is kind of an interesting association because it raises the question, is insulin use a proxy for duration or severity of diabetes, or is insulin use maybe giving us another clue potentially about the underlying pathophysiology? And I will hope during the talk to show you some data that suggests that hyperinsulinemia itself might potentially contribute to the increased risk of heart failure which occurs in the context of diabetes. Here's a recent study that was recently published by Justin Echufo-Chugai, currently at the Brigham and Women's Hospital, really just looking at hospitalizations for heart failure and asking what percentage of people with heart failure diagnoses are diabetic. Remember, in the population, it's 10 to 15%, but in the heart failure population, it's about 50% of individuals with heart failure are diabetic. And in fact, if you look at the flavor of heart failure, whether it's heart failure with reduced ejection fraction, borderline ejection fraction, or preserved ejection fraction, there is significant enrichment in those populations with individuals who are diabetic. Now, heart failure, we need a heart failure moonshot, because if you look at this slide, cancer, 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 heart failure. So it turns out that the mortality of heart failure is actually significantly worse than many cancers. And if you are diabetic, your prognosis is even worse than if you are not. And let me give you 2 examples of that. This is another paper from Justin's group, just looking at people with advanced heart failure who required resynchronization therapy or an implantable defibrillator, and just looking at their survival. And you can see in the blue that if you are diabetic, your chance of survival was significantly worse than if you were non-diabetic. So it raises the question then, why is this? And so clearly, one of the obvious things to look at is glycemic control. So if your glycemic control is less robust, are you at greater risk of heart failure? And in fact, this meta-analysis would suggest, in fact, that this is the case, that if you have an increase in hemoglobin A1C, with the exception of this one study, that in fact your risk for heart failure is amplified. So if glycemic control amplifies the risk for heart failure, then the question becomes, does improved glycemic control reduce your risk for heart failure? And so the question then is, what is the impact of glycemic control on the outcome? Before I show you the data in heart failure, let me remind you of a number of landmark trials, some of which were funded by the NIH, that looked at the impact of tight glycemic control on a number of cardiovascular endpoints. And this was a big wake-up call for many of us in the field, because in all of these trials that looked at a variety of ways of achieving tight control in individuals with type 2 diabetes, there are a couple interesting messages. One, people weren't necessarily living longer. And secondly, in some studies, not all, it appeared that maybe there was an increased risk of cardiovascular events. Now, if one looks at the relationship between diabetes therapies and heart failure, the picture is somewhat mixed. That some medications, looking across a broad swath of epidemiological literature, would suggest that if you are on these medicines, that they either could potentially exacerbate or increase your risk for heart failure, whereas a few others, including new agents such as SGLT2 inhibitors, and of course the standby metformin, may actually be somewhat beneficial in the context of heart failure. Let me give you a couple examples of this from the literature. So this was an analysis of those same tide control trials I just mentioned earlier, looking at the impact of any of these trials on the risk for heart failure. And the way that you look at these data is that if, in fact, tide control was beneficial, then you would be on this side. If tide control was less beneficial, you'd be on this side. Most studies are kind of to the left of this line, again, suggesting that there's something about intensive control that may not necessarily be good for the failing heart. Here's another example. This was a SAVOR-TIMI trial looking at a DPP-4 inhibitor, saxagliptin, and one of the signals that they saw early in this trial was that in the green were the patients randomized to saxagliptin relative to placebo, and these were looking at the hospitalizations for heart failure, and you can see that there was actually a significant increase in heart failure hospitalizations in those people who were randomized to the DPP-4 inhibitor. This was a drug that never quite made it to market. So in concept, this was a good drug. It was a dual PPAR alpha, PPAR gamma agonist. So it would treat dyslipidemia, also have a glycemic benefit. But the trial was stopped because people were actually having adverse events, including increased hospitalization for heart failure. And then this is another interesting study, which is done a slightly different way. So what Ken Margulis did in this study was he took people with heart failure, stage 3 or stage 4, American Heart Association heart failure, and then randomized them to be treated with a GLP-1 receptor agonist, liraglutide, or not, and very efficacious anti-hyperglycemic medication, and in fact, in some randomized trials, will reduce certain macrovascular cardiovascular outcomes. But in this very specific population of patients with heart failure, not a big study, but what he noted was that the death rehospitalization for heart failure, or ER visit for heart failure, was actually statistically higher in the liraglutide group relative to the placebo group. And then when he did a subgroup analysis looking at who were the ones driving this association, in fact, it was the patients with diabetes, but not the patients without diabetes. So I'm painting you this picture that things which lower blood glucose very well might not necessarily have a proportional benefit in terms of heart failure outcomes. And then this study came up, so SGLT2 inhibitors, which kind of continues to rock the world because nobody knows why it happened or how it happened, but this is what happened. So you give a drug that makes you actually pee out more glucose, right? It inhibits sodium glucose transporters in your kidney, and you had significant reduction in mortality, a significant reduction in the heart failure hospitalization, and this, in fact, might be a class effect because another study using another drug called canagliflozin pretty much showed very, very similar results. So you have a scenario where clearly lowering blood glucose may not be enough, and there might be some other attributes of our therapies that might be important if one wants to then tackle this issue of heart failure. So why have we been missing the mark is the question. And I think that one of the reasons for that, which was attempted to be summarized by Deepak Bhatt here in this cartoon, was that, well, maybe if you look at anti-hyperglycemic therapies in people with heart failure, it is possible that it has nothing to do with the therapy. It's just chance, it's a randomized trial, and it's the luck of the draw, and maybe it's just bad. Although when it happens for multiple classes of agents, it makes it perhaps a little bit less likely that that's what's happening. Or it could be something as simply that the agent is just putting more volume on. And that's certainly true for thiazolidinediones, TZDs, but not necessarily all classes. Or potentially there might be direct myocardial effects of these agents. And then I'd posit that there might be something else. And I'd like to kind of introduce you to this concept that there might be a cardiomyopathy of obesity and diabetes. And the way that we have defined this over the years is that really it represents an intrinsic defect in cardiac muscle that increases the likelihood that cardiac dysfunction will develop in the context of additional stressors such as ischemia or cardiac hypertrophy. Now, what's the evidence for this? This is a wonderful study done by Elizabeth Selvin and colleagues at Hopkins, where they have for many years been studying this cohort in the ARIC study, looking at people in the community and looking at risk factors for atherosclerosis and cardiovascular disease. And one of the nice things about epidemiology is that when you have samples in the freezer, as technology advances, you can go back to the freezer and learn new things. And in this case, they applied a very highly sensitive troponin assay to blood samples of individuals who were stratified by diagnosis, so either non-diabetic, pre-diabetic, or with diagnosed diabetes. And you can see very clearly that there is a significant, almost linear increase in the amount of this highly sensitive troponin that was detectable as you went from non-diabetes to diabetes, suggesting that there is subclinical myocardial injury. Remember that this is not the level that you see in the ER when you have a heart attack. That's up here somewhere. But this is just really just saying that there is evidence of subclinical muscle damage that's occurring. And more interestingly, when she then looked at people, so she made this arbitrary cutoff of 1.4 below or 1.4 above. And even with that cutoff, there was still this linear relationship, both in terms of the risk of any cardiovascular event or death, or a risk of heart failure that was very proportional to the level of subclinical myocardial damage that was occurring in these individuals with diabetes. And of course, amplified, of course, in those who were on the right side of this cutoff. So it really, comes to this notion then is what is happening in the heart in the context of diabetes. And so I've put up this cartoon from a review that we published a few years ago that really attempted to capture what was known or what was being studied. And you can see everything is being studied. And it's kind of like insulin resistance, right? There are about 50 different pathways that might lead to insulin resistance. And unfortunately, in the context of diabetes, it really is 1,000 flogs on the heart in this abnormal metabolic milieu, with strong evidence implicating many of these pathways in the pathophysiology of diabetic cardiomyopathy. So time doesn't allow me to go through all of these, but I'm going to pick a couple from our own lab just to give you some insight into some of the mechanisms that we and others have identified. So we'll first talk about work in the mitochondria and the concept of lipotoxicity and increased fatty acid utilization, which occurs in the heart in diabetes. So let me just give you the headline. The headline is this, that the heart is a substrate-hungry muscle. It doesn't have the option to fatigue or rest, and it really needs to generate anywhere from 6 to 9 kilograms of ATP per day to maintain its contractile function. And as a result of that, is an avid user of multiple substrates. But what happens in the context of diabetes is that there is a switch of the heart which tends to like to burn fat, but it tends actually burns more fat in the context of diabetes, and somewhat at the expense of using glucose. And this occurs in concert with mitochondrial dysfunction. So let me show you some data from a human study that was done by Ethan Anderson when he was at East Carolina University. And this is a nice example of translational research because he told his cardiothoracic surgeons that when you go in to do coronary bypass surgery and you cut off the atrial appendage, rather than send it to pathology, send it to me. And what he showed was that if you look at diabetics, they had more triglycerides in their atrial appendages, which correlated very nicely with their hemoglobin A1C. And then he permeabilized the muscle and did mitochondrial respirometry on these. And you can see that the diabetics in the black had impaired mitochondrial respirations, decreased odor consumption, that also correlated with A1C. So the more diabetic you were, the lower your mitochondrial respiration was. He also then measured hydrogen peroxide emission, so superoxide, a proxy of superoxide production by these mitochondria. And you could again see very clearly that the diabetic individuals made more hydrogen peroxide, shown here as well with different substrates. And if you looked at markers of oxidative damage in these tissues, again, this was increased in the diabetics. So here is a Western blot showing hydroxynoninol and adduct, which was increased in the diabetics. Here is a 3-nitrotyrosine Western blot, again, showing that that was increased in diabetics. We have done a lot of work in animal models, and we started off looking at these very dramatic genetic models of obesity and type 2 diabetes, and began to look at their mitochondria. And again, we saw that the mitochondria were quite dysmorphic, and there was lipid accumulation in the hearts of these animals. And to sort of summarize what happened to cardiac metabolism in these hearts when we perfused them, in fact, we saw that there was this switch in substrate use, that the hearts were using much more fatty acids, shown here in black for the diabetic animals, and were oxidizing less glucose and having less glycolysis. Now, one of the things that happens when you burn more fat is that you actually use more oxygen to make your ATP. And therefore, when we actually measured oxygen consumption in these hearts, we saw an increase in O2 consumption despite a reduction in cardiac power. So if you think about it as like a marathon runner, if you're a marathon runner, and I'm sure this room is full of marathon runners, and you're not looking at one here, so I could be the control, that if you actually put me in a box and measured my O2 consumption after I ran maybe 2 or 3 miles compared to the marathon runner, he or she would use significantly less oxygen than I would. So that person is very efficient. The diabetic heart is the anti-marathon heart in the sense that they're rather inefficient. So they have increased oxygen consumption rates, even although they might be doing less work. And so one of the questions then that we asked was, is it simply because they're burning more fat? And the answer is no, because in fact, in addition to using more fat, what happens at the level of the mitochondria is that they hit on a mitochondria that has significant impairment in the machinery of the electron transport chain. And as a consequence of that, there is a propensity to over-generate reactive oxygen species or superoxide. And an important consequence of superoxide overgeneration is that superoxides will actually lead to a phenomenon in mitochondria called uncoupling. So this is the biochemistry part of the talk, and this is a schematized mitochondria, and substrates are coming in. Electrons are pumped across the electron transport chain, and I apologize to Dr. Balaban, who's in the audience, because he knows that the electron transport chain looks nothing like that in reality. But from the standpoint of a cartoon, it works for me. But essentially, as the electrons are being pumped across the electron transport chain, they do generate superoxide. And it turns out that superoxide activates proteins within the mitochondrial membrane called uncoupling proteins that then will cause protons to leak back into the mitochondrial matrix. Now remember that when protons enter the matrix, whether through ATP synthase or through these uncoupling proteins, they interact with molecular oxygen to produce water. So if your mitochondria is completely uncoupled, you can have a very high oxygen consumption rate and are not really making ATP. So what we others have shown is that in the context of diabetes, there is uncoupling of the mitochondria, which also drives this increase in oxygen consumption rates and reduces the amount of ATP energy that these mitochondria are generating, which we think is an important mechanism for this decrease in efficiency in the heart in the context of diabetes. Now, this is also true in people, and I'm going to go back and forth between humans and mice because I think it's important to really validate some of these observations. So this is work from Linda Peterson at WashU, and she took a cohort of women with obesity and type 2 diabetes and used a PET approach to measure uptake of fatty acids or oxidation of fatty acids. And this is showing in relationship to glucose tolerance. So as you become more diabetic, the heart uses more fatty acids. But importantly, she also showed very nicely a nice relationship between obesity as a proxy for insulin resistance and myocardial oxygen consumption rate, as well as myocardial efficiency. So the first message then is that if you're diabetic, the heart is using more fat, it's using more oxygen, it's less efficient. And therefore, if you then start to get ischemic, then you're much more likely to get to that point of injury earlier because your oxygen requirements are that much more high. Now, the other thing I just want to give you a little flavor of is, what else is this ROS doing to mitochondria besides uncoupling? And I'm going to share with you just a little snippet of a recent publication from our lab where we really showed that ROS also was affecting mitochondrial form and function. So for those of you who don't think about this a lot, mitochondria, were perhaps at one point bacteria and still do things that bacteria like to do, which is to fuse and share DNA and to divide and to fuse and share DNA. And in a sense, that's what all of life does from time to time. And so this process is called mitochondrial dynamics, where the mitochondria will either undergo this process of fission or a process of fusion, and there are a number of molecules well established to drive that process. So we made an animal model of increased fatty acid utilization by overexpression of an enzyme, acyl-CoA synthase. And this is a PET scan of a mouse, and you can see increased fatty acid uptake in that heart relative to the control, and this was about at the level of what we'd see in diabetes. And what was very interesting in these animals was that when we looked at the mitochondria of these animals, there was a really very dramatic kind of change in the mitochondrial morphology, as shown here. Now, for those of us who look at EMs, you live in this 2D world. So you make a section and you look down on your image and you see what the mitochondria look like. And so initially we said, oh my gosh, look at this. These mitochondria are actually completely fragmented. We submit the paper and the reviewer said, we want more. I'm sure this is something that's very familiar to many of you in this room. And so they really weren't convinced that they were fragmented. And so they forced us to actually put on a virtual reality headset and look at a 3D reconstruction of what was happening to the mitochondrial network. And that's depicted here. So this is normal. And if you look here, you'll see that this is what the lipids are doing to the mitochondria in the animal model. So it's really becoming almost like spaghetti or noodles and going in and out of the plane. So when you did a section, you saw what appeared to be mitochondrial fragments, when in fact you were seeing a mitochondria that was really stretched and twisted and tortuous and not functioning very well. And so it turns out that what's driving this morphological change is superoxide. And to kind of show that much more directly in the same paper, we looked at animals that overexpressed superoxide dismutase, so it's a ROS scavenger. And then we crossed these animals with our transgenic mice, and then we had some compound mutants. The key point I want to make out here is this, that if you scavenge ROS aggressively in this transgenic heart overexpressing superoxide dismutase, you get large mitochondria, which is an opposite to what we saw when we were overproducing ROS by increasing fatty acid use. And this is shown here in terms of mitochondrial size in this SOD2 transgenic animal, comparing this with the ACS mouse that has increased ROS. And then, of course, when you put the two together, you can normalize mitochondrial size. So mitochondria are actually not these static organelles that you see on these EMs, but they're really dynamically being regulated by the energy that it's receiving in a superoxide-dependent manner. And we went on to show mechanistically that the ROS was driving this process by various modifications on critical molecules that were involved in this mitochondrial dynamics process. So this has been picked up by various people, and I got this off the internet because Elizabeth Dauman did a piece on our paper and had this very nice cartoon which essentially had the tagline that increased myocardial fatty acids utilization potentially could amplify the risk of heart failure and diabetes through a number of mechanisms, including alterations in mitochondrial structure and mitochondrial function. Okay, so that's a little bit on mitochondria and lipids, and I'm going to give you a little bit on insulin signaling. So I'm going to give you some evidence that we think that hyperinsulinemia may also be contributing to the increased risk of heart failure which occurs in diabetes. So this is my second biochemistry slide. So I apologize for everybody in this room who studies insulin action. This is a very simple representation of the insulin signaling pathway, but again, it works for the purpose of this presentation. So insulin is a tyrosine kinase, a cousin of the IGF-1 receptor, and when it binds this ligand, activates a number of intracellular molecules to transmit its signal into multiple pathways summarized here, including metabolic pathways, cell survival, and cell growth pathways, as shown here. And I'm going to show you some data on some important intermediates in the insulin signaling pathway, such as AKT, IRS proteins, and the glucose transporter GLUT4. And so when you think about insulin resistance, we think about a hormone that normally rises in response to feeding with the primary goal of maintaining homeostasis to push glucose back into muscle, suppress hepatic glucose production, and to suppress lipolysis. And in insulin-resistant states, these fundamental pathways are impaired, leading to a metabolic milieu that is characterized by hyperinsulinemia, increased levels of free fatty acids, and ultimately increased levels of glucose. It turns out that if you measure these same things in people with heart failure, you also see that heart failure itself is an insulin-resistant state, with mechanisms that are very similar to what we see in diabetes, such as increased ectopic accumulation of lipid in muscle and liver, increased inflammation in skeletal muscle, increased inflammation in adipose tissue, for example. So the question then is, what happens to insulin signaling in the heart in the context of diabetes or in the context of heart failure? So the endocrine cognizant in this room know very well that in insulin-resistant states, if you measure insulin action in skeletal muscle or in adipose tissue or in the liver, you'll see that insulin action is impaired. But we didn't really know whether that was also true in the hearts. And this is a very interesting study that was done by Stuart Cook when he was at the Hammersmith in London, where he took patients in the cardiology clinic and said, sign this consent form. This is for your treatment, and then this is for a pretty intensive clinical protocol, which I'll describe in a second. These are people who were coming through the clinic and were undergoing coronary angiography because they had a stress test that they failed, or there were patients with heart failure, secondary primarily to valve disease, who were coming in for surgery. And basically, he convinced them to have a glucose clamp, a skeletal muscle biopsy, a PET scan, and then when they went to the OR for their coronary artery bypass surgery, they had a left ventricular biopsy as well. The IRB approved the study. At least that's what, as best I could tell. But importantly, it was really very informative. So let me tell you who these patients were. So these are the controls. These are the heart failure patients, LVD. These are the type 2 diabetics here. You can see that the type 2 diabetics had normal LV function as measured by ejection fraction. The heart failure patients is heart failure with reduced ejection fraction. So those are your three cohorts. And importantly for the clinicians in the audience, if you look at the hemoglobin A1C in the diabetic patients, their A1Cs were 6.6. So I guess that's the NHS at work, right? So when they did skeletal muscle biopsies of these individuals, what they actually, well, before they do that, let me just mention the glucose clamp. So this is the glucose clamp looking at whole body glucose utilization. You can see that the heart failure patients or the diabetic subjects were insulin resistant. So they had decreased whole body use of glucose. And that was also true in the heart. This is the skeletal muscle biopsy result. So not unexpected, when they did the skeletal muscle biopsy, whether in the heart failure patients or in the patients with type 2 diabetes, there was, during the glucose clamp, there was significant impairment in the phosphorylation of IRS1, again, a sine qua non for insulin resistance in skeletal muscle. But in the heart, it was actually unexpected. In contrast to skeletal muscle, where insulin signaling was reduced, in the heart, insulin signaling was increased. So these dots are PI3 kinase activity, and quantified here. And you can see that in patients with heart failure or type 2 diabetes, there was increase in the activation of PI3 kinase that was associated with IRS1. when you do a regression of whole body glucose disposal, so the higher the number, the more insulin sensitive you are, the lower the number, the more insulin resistant you are, you can see that patients who were most insulin resistant had the highest activity levels of PI3 kinase in their hearts. And this is also true in terms of hyperinsulinemia. The most hyperinsulinemic patients had the highest levels of PI3 kinase activity. And then downstream, another signaling intermediate, GSK3 beta, the phosphorylation was higher in the hearts of individuals with diabetes. So you have patients who have decreased glucose uptake in their heart, but increased proximal signaling from the receptor through to PI3 kinase. So why is the glucose uptake lower? So it turns out that they actually had less GLUT4 on their cell surface. So what did we learn from this? We learned that in two conditions, whether heart failure or type 2 diabetes, that is characterized by generalized whole body insulin resistance and hyperinsulinemia in humans, there's a more selective insulin resistance in the heart, characterized by decreased insulin-mediated glucose uptake, but with proximal signaling to PI3 kinase and AKT. So is this true in mice? So we did a study where we put animals on a Western diet, which mice really like. One of my postdocs one day came to my office with 2 pellets of animal chow. One was a Western diet, and one was the cardboard pellet that mice are given this hardship to eat. And he said, Dale, I actually tasted this. And this stuff is actually really nice, right? And so these animals, put them on this Western diet, and they will chow down and will gain weight and become insulin resistant. So we wanted to look very early on, within the first 2 weeks. And after 2 weeks on this diet, they got a little hyperinsulinemic, so they doubled their insulin levels. But at this point, their lipids weren't so abnormal. And this work was done by Jordan Wright, who at the time was an undergraduate student, who then went off to Michigan with an MD, Ph.D., and is now a PhD resident at Vanderbilt. So as we were talking earlier about the pipeline, this is a nice example of somebody who you caught very early as an undergraduate who is now, a physician scientist. So Jordan measured insulin-stimulated glucose uptake in cardiomyocytes from these animals. And in fact, he said, they look very insulin resistant because, as you can see in the black bars, that the increase in glucose is significantly lower or impaired compared to the white bars or the controls when we expose them to insulin. So then he did this experiment where he gave the mice insulin and then measured the phosphorylation of AKT. And this is when you get the distress telephone call. It's like, something is wrong. I've done it 3 times. It makes no sense. They don't transport glucose, but there's way more AKT activation that's taking place. Sorry, this is a little fast, this clicker, right? There's way more AKT activation in these hearts relative to the controls. And I said, okay, Jordan, this is interesting. So why are they not transporting glucose? And it turns out that, like the people I just talked to you about, they also didn't have enough GLUT4 on their membranes. So that's nice because I now have an animal model in which I can now begin to ask some additional questions. Again, to remind you, is this concept of a condition of generalized insulin resistance where the heart, however, is a little bit selective. There's decreased glucose uptake, but there's intact or increased signaling from insulin to PI3 kinase to AKT. So the question then that we asked was, can hyperinsulinemia or increased insulin signaling exacerbate heart failure? And to do this, we took advantage of a model that we can use in mice called transverse aortic constriction, where you put a little knot around the aorta, you narrow the lumen, the heart hypertrophies, and ultimately the heart fails. And one of the very interesting observations that we made in the study was that if you do transverse aortic constriction after 2 weeks, there is increased activation of insulin signaling pathways. So there's increased phosphorylation of IRS1, increased activation of AKT. That's quantified here. More intriguingly, if you took a cardiomyocyte and just isolated it from a heart in a dish and simply stretched it, there was also increased activation of IRS signaling pathways. So there's something about cardiac muscle and stretch which is activating insulin signaling pathways. And when these animals develop heart failure, they also develop whole body altered metabolism, as evidenced by hyperglycemia, hyperinsulinemia. And if you look for insulin signaling in a peripheral organ, in this case the liver, their livers were insulin resistant. So you have hyperactivation of the signal in the heart, but insulin resistance in the periphery. So we asked the question then, would reducing insulin signaling ameliorate left ventricular dysfunction following aortic constriction in this model? And so what we did was our lab had generated some mice where we could inactivate or reduce the expression of insulin receptors selectively in cardiomyocytes. So these are what we call these circle mice for cardiomyocyte insulin receptor knockout. So we studied the heterozygotes for about a 50% reduction in insulin receptors and repeated the experiment. And you can see that these animals did not hyperactivate IRS1 after transverse aortic constriction, nor did they activate AKT, that's quantified here. And more importantly, they didn't hypertrophy as much, and they maintained their fractional shortening. As a measure of left ventricular function in the mouse, by echocardiography, you can see that the control animals, after transverse aortic constriction, reduced their fractional shortening. This was preserved in those animals in which we reduced insulin signaling. And in fact, there's also less apoptosis shown here. So this suggested to us that in the context of this particular model of heart failure, which is pressure overload-induced cardiac hypertrophy, that hyperinsulinemia and myocyte stretch can both independently activate IRS1 and increase the signaling through AKT and contribute to heart failure by increasing left ventricular remodeling. So in biology and in life, many things come in pairs, and there are actually a number of IRS isoforms, IRS1, IRS2, which are expressed in the heart, and there are a number of AKT isoforms which are expressed in the heart. So we were wondering whether both IRS isoforms were contributing to this relationship between hyperinsulinemia and heart failure. So to address this, we made animal models where we selectively deleted either IRS1 or IRS2 in the heart, and then subjected them to pressure overload hypertrophy. So what you can see here, and you just maybe focus here on this histology slide, is that if you go comparing a sham heart to a heart after transverse aortic constriction, this is 4 weeks, you see evidence of increased fibrosis and injury in the heart. This is also true in animals that lack IRS2, but importantly, animals that were lacking IRS1 in the hearts were completely protected from that phenomenon. And the other thing that we then also showed was that this injury pathway was also selective at the level of AKT, specifically for AKT1, but not AKT2. That's a busy slide here, but this is a quantification shown here that in the controls after TAC, AKT1 is hyperphosphorylated, IRS2 knockouts the same, IRS1 knockouts not, and no change in AKT2. So is this true in human hearts? And the answer is yes. So we got some left ventricular assist device samples from the cardiac surgeons and just looked at IRS1 phosphorylation or AKT1 phosphorylation or IRS2 phosphorylation or AKT2 phosphorylation in these human hearts of individuals with heart failure. And again, you can see this selective activation of IRS1 and AKT1. So what this has really taught us, then we believe, is that hyperinsulinemia is somehow potentially exacerbating heart failure by a signaling pathway that involves specifically IRS1, but not IRS2, and AKT1, but not AKT2. And in terms of mechanisms, of which there are many, I'm just going to show you one mechanism, that these IRS1 deficient hearts have an increased amount of somewhat cytoprotective nucleotide, sadly GNP, in their hearts. And we're now pursuing the mechanisms linking those two signaling pathways. So I want to leave you with the idea then that increased insulin signaling in the heart, we think, actually is growth promoting and may actually accelerate left ventricular remodeling. And remember that type 2 diabetes, for the most part, is a profoundly hyperinsulinemic state. Now, there's another insulin receptor story that we also recently discovered, and it's this. So again, you high-fat feed animals, they get diabetic, they get hyperinsulinemic, and if you do it long enough, their hearts will fail. So their hearts will dilate, they will get fibrosis shown here by these blue stains, and their ejection fractions will fall as a function of time. And this is a collaboration with Kevin Jiang at UC Davis. And Kevin is a beta adrenergic receptor biologist. And what we ultimately showed was that the insulin receptor actually exists physically in a complex with the beta-2 adrenergic receptor. And when there is hyperinsulinemia, this complex actually becomes stronger, and activates another signaling pathway involving a kinase called GRK2 through beta-arrestins to another kinase called ERK to induce the expression of a phosphodiesterase called PDE4, which then degrades cyclic AMP. And here is some evidence for that. If you knock out insulin receptors, you have less PDE4. If you high-fat feed, PDE4 goes up. If you high-fat feed an insulin receptor deficient mouse, you don't see this increase in PDE4. So what we then did in the study was we actually then essentially tried to block this process both pharmacologically and genetically to see if we could preserve cardiac structure and cardiac function. So we had mutant mice that were lacking beta-2 receptors, or we were able to either genetically or pharmacologically inhibit GRK2 or beta-arrestin. And this is kind of shown here that in the control animals on a high-fat diet, they lose ejection fraction, but in knockouts for beta-2 receptors or beta-arrestin 2, they maintain their ejection fraction. We also used a couple of drugs. We used a beta-1, beta-2 blocker, or a GRK2 inhibitor at high doses. So paroxetine is an antidepressant, so we weren't using it at antidepressant doses. These are higher doses that block GRK2, again, as a proof of concept. And again, we took the animals to a point where they developed LV dysfunction and then treated them. And again, you can see that the animals treated with either paroxetine in red or carvedilol in blue, again, had a restoration of their ejection fraction, despite no real change in the degree of hyperglycemia or the degree of hyperinsulinemia. So this then suggests to us that taking both sets of data together, that early on in the insulin-resistant state, we think that hyperinsulinemia drives signaling pathways that drive aberrant cardiac hypertrophy and other pathways summarized here that can increase left ventricular remodeling, and that further on, as it becomes more protracted, there's this GRK2 mechanism which also may contribute to cardiac decompensation. So we believe that these data would suggest that in the context of heart failure, we think it's imperative to identify novel ways to manage diabetics, particularly if they're hyperinsulinemic, to optimize metabolic control at the lowest possible levels of systemic insulin. And secondly, it really is imperative now to pursue additional mechanisms that we might use to therapeutically limit the indirect effects of hyperinsulinemic signaling on the failing myocardium. So this talk was entitled Sugar and the Beating Heart, and I actually haven't said much about sugar. And so I'm going to end with just a couple of sugar slides, just to tell you a little bit of new work that we are doing. So we have got access now to a very robust heart failure tissue bank, and have been doing some metabolomics profiling on those samples. This is a collaboration with Ken Margulies at the University of Pennsylvania. And we're particularly interested in looking at individuals who are stratified either as donors or heart failure patients, either obese or diabetic or not. And so this is their ages. They're between 40 and 60. You can see in the non-failing cohort, so these are donor hearts, we have individuals who are obese, individuals who are, I guess, less obese, and then we have patients with heart failure. And again, this is heart failure with reduced ejection fraction, as shown here. And again, by virtue of the nature of these samples, we don't have A1Cs, what we can measure like fructosamine as a proxy of glycemic control. You can see that the diabetics have higher fructosamines, and the obese subjects are tending towards higher fructosamine levels. So let me give you a little insight into some early metabolomic data that we have observed. So a very striking phenomenon that we have seen in these hearts is what I call a glycolytic backup. So accumulation of This is free glucose. This is glycerol-3 phosphate. These are our mitochondrial metabolites. And so it appears that things are backing up in glycolysis and spilling over into some side pathways, as shown here. So galactose, galactosamine, mannose, sorbitol. And you say, well, why are we interested in these things? Well, these things actually activate tissue-damaging signaling pathways that I've attempted to summarize on this slide. So mannose will increase end-linked glycation, increase extracellular matrix remodeling, and increase inflammatory signaling. Sorbitol, of course, through the polyol pathway, will lead to depletion of NADPH and glutathione, leading to oxidative stress. And then this pathway will actually lead to increased advanced glycation end products, rate signaling, and inflammation. So given that we had no evidence of a glycolytic backup, we said, can we model a glycolytic backup in a mouse heart? And so we did this by inactivating a recently identified pyruvate channel called the mitochondrial pyruvate transporter. And we predicted that this would give us a nice model of glycolytic backup, and then what could we learn about that and left ventricular remodeling. Very nice for us and for this story is that these animals actually recapitulate the metabolic phenotype in a diabetic heart, in the sense that they have increased fat oxidation and decreased glucose oxidation and increased myocardial oxygen consumption. And if you follow these animals over time, they fail, which is nice for this story. They get dilated and they have a reduction in their ejection fraction. So what have we begun to learn from these animals? So we have now landed on this cancer metabolite called 2-hydroxyglutarate. And so if anyone in this room studies cancer metabolism, this is a very important metabolite because it drives a lot of oncogenesis through multiple mechanisms, including, for example, stabilization of HIF-1 alpha. It turns out that these animals accumulate 2-hydroxyglutarate, and I don't have time to tell you how we have manipulated this, but one of the things that Heinrich Teitmeyer has shown very nicely recently is that 2-hydroxyglutarate can have profound effects on cardiac metabolism, in fact, sort of reversing some aspects of the TCA cycle and leading to accumulation of metabolites that ultimately lead to histone modifications and alterations in epigenetics and alteration in gene expression. So I'm now beginning to think that heart failure is probably going to be like, cancer of the heart. Has a terrible prognosis, but now that changes in metabolism might also be driving some of the aberrant signaling pathways that we see. So glucose metabolism and heart failure, I believe, is no longer just simply a matter of energy production, but I think our recent work and those of Heinrich Teitmeyer really reveals a new frontier of metabolites as molecular signals. And I think that as we begin to sort of understand what's happening at the level of pathophysiology, we need to begin to understand how these metabolites are leading to aberrant signaling pathways, which I believe ultimately can be therapeutically targeted. So I hope I've tried in the last 45 minutes or so to show you that we are confused, that we really don't know enough about what's happening in the context of diabetes, which is why I had made this title, Sugar and the Beating Heart and the Conundrum of Heart Failure in Diabetes. And my hope is that I've given you a little flavor of some of the complexities that are linking this very prevalent metabolic disorder with an increased risk of heart failure. So I'm here because they are there, and this is a picture of my lab. Kensuke Tsushima did the work on the mitochondrial remodeling in relationship to lipids. Yuan Zhang is working on the pyruvate carrier story, and Eric Weatherford continues to work on the insulin signaling story. And some of the published work I showed you before was really done by other members of the lab, many of whom have now gone on to run their own labs. And it takes a team to do this work, and many collaborators have contributed to these studies. And I continue to be indebted to the NIH for keeping us going through thick and thin and through many A1s and A2s. So with that, I thank you very much for the opportunity to share this with you this afternoon.
thank you for a really interesting romp through a lot of data and interesting conclusions, and as you say, much of things that we might continue to talk about even now. So there are microphones in the aisles for people who might want to pose a question. Please do so. We can start right over here. Yes, sir.
Whenever you see a type 2 diabetes, you know that you're going to have obesity, metabolic syndrome as well, kind of correlated. And then the big boy on the block now, non-alcoholic fatty liver disease, kind of comes in. I'm wondering, there already is one FDA-approved drug for non-alcoholic fatty liver disease, that is non-alcoholic hepatosteatosis. Is there any correlation with this with your heart failure, number one? And number two, would it be worthwhile comparing trials for the drugs that's now used for non-alcoholic fatty liver disease?
Right. So I'm not aware of any trial that's looking specifically at the link between treating NAFLD and heart failure. What I can say is that there are groups that have looked at measuring both liver fat and heart fat in people at risk. And certainly, if you have NASH, then you're much more likely to have changes within your heart that may contribute to the increased risk of heart failure. But studies directly linking both, as far as I'm aware, are yet to be done.
Okay, thank you.
Thanks. Over here.
Well, congratulations for covering complex issues. Being a diabetic, obviously, I'm interested of the too many implications of too many partners. So one of the issues I thought I could do without too much medication, other things, is exercise. So this is a major effect, and you could put some of the mice, even some of the patients, and see what is the net outcome of all the complications that is happening.
Yes, so you make a very, very important point. And yes, exercise is important. There's a particular kind of heart failure called heart failure with preserved ejection fraction that's very prevalent in individuals with type 2 diabetes and the metabolic syndrome. And certainly one of the most efficacious things in those patients in which there really aren't very good drugs is exercise. The challenge, of course, is that if you have heart failure, your ability to exercise is diminished, which therefore means that ultimately we have to find other targets that we can mimic exercise with. I see Bob is exercising his finger, right? In order to see some benefit.
Obviously, the intervention in the early stage is critical.
Absolutely.
Thank you.
I would agree with that.
Over here on the right.
Thank you very much. Very interesting. One of the first slides that you presented showed that the risk of heart failure is much greater in diabetic women compared to men.
That's correct.
And I wondered if you conducted the experiments in males or females, and if both, do you see the differences in mechanisms?
Yeah, that's a very good question. So there's someone in the audience, Dr. Tish Murphy, who knows a whole lot about gender and sex and heart failure than I do. Not many studies in animal models have been done. There have been some really nice human studies by Linda Peterson's group at WashU in humans, male and female with and without diabetes. And clearly what she has shown is that the patterns of substrate metabolism, which is what she studies, are certainly different between males and females. So that's at least giving us a hint. But I think there's a lot more that we need to do, particularly with regards to some of the other mechanisms that I showed in the summary.
Great. I know our speaker has to catch a plane, but we can take two more quick questions. Yeah. Phil.
Very quick. That was a beautiful talk.
Thank you.
So after the DCCT, it was clear that better insulinization had better outcomes for microvascular disease. Now,
that's correct.
You're raising a question about whether that might be a bit of a contradiction for macrovascular disease. So first question is, how should we translate this to a community? And the second question is, how much glucose does the heart use anyway?
Right. So let me answer the second question first. So the heart uses some glucose. I'm not going to give you in terms of grams and kilograms and whatnot. But if you look at the proportionate use of, say, fat, lactate, and glucose, I would say under normal, healthy circumstances, it's probably getting about 50 to 60% of its ATP from fat, probably about 20% from lactate, and the rest from glucose. So it does need glucose, not just only for energy, but for things like anaplerosis, etc. If you completely constrict glucose use, at least in animal models, the hearts aren't very happy. So you do need some glucose. The question then is, how much is too much? And I think that The complication in the heart and diabetes is twofold. Number one, that it ends up using more fat. And then secondly, because of the mitochondrial defect, you get accumulation, I believe, of these kind of glycolytic intermediates, which can then have an impact. Now, to your earlier question, which is how do we translate this into practice? And what I would say at this stage is that we really have to sort of personalize our management of many of our patients. So I think one of the things as a broad theme in diabetes in general is that, not everybody should have their A1C driven down to 6, particularly if you're elderly and at risk of hyperglycemia, etc. I think we all accept that. I'd also argue that in the context of heart failure, we really have to balance our therapeutic options very, very carefully. Certainly, the heart failure has to be optimally managed. I think we need to try to manage the metabolic abnormalities in a way that I would argue should promote less hyperinsulinemia if possible, should not increase volume if possible. And so that does limit. I know, for example, in the cardiovascular community, everybody's jumping on top of SGLT2 inhibitors right now in high-risk patients. I would have a caution that it's probably a little bit early. Those are very narrow studies and very, very predefined populations. I'm not sure yet if it's generalizable. I know that there are clinical trials underway right now in heart failure patients, for example, to see if SGLT2 inhibitors will have an impact on the outcome. I think the jury is still out. We need to wait on those data.
Thank you.
Thank you. Last question.
Dale, thanks for that outstanding talk. So we've seen flexibility, at least in healthy subjects, in terms of fuel utilization, whether fatty acid or glucose uptake. Do you think, based on the model that you've developed, if you could shift the heart towards increased glucose utilization, there would be less generation of the ROS and less likelihood for heart failure, or will the hyperglycemia lead to the same kinds of problems?
Sure. So like everything in life, there's a balance. And didn't have time to show this, but we have done those experiments. So we have mouse models where we can actually inducibly increase the expression of glucose transporters. And what we see in the context of the pressure overloaded model is we do see less remodeling. With that said, if we turned up too much, we then see glucose injury. So I agree with you that having the metabolic flexibility is important. It's now how does one kind of tune that, in the clinical context, and that I think is a challenge.
Well, you've given us a wonderful example of a lot of wonderful basic science that also translates into really practical clinical implications. If we could just figure out exactly how to put that all together,
right.
And you've gotten us well along the way. So
keep
please,
funding me.
I see. You left the door open to more work needs to be done, as they all do. Well, let's thank our speaker again for a wonderful presentation.
Thank you.